and you find associations with them, we know they're not independent. Everything interacts with everything. Detect interaction between loci, therefore it doesn't exist, which is absurd. Of course it exists. And of course all these estimates of effect size in these polygenic risk scores are nonsense for this reason. So what I want to do is, this is another paper. I just want to go through this because the title is hilarious. Genetic variants in novel pathways influence blood pressure and cardiovascular disease risk. So you'd think they found variants that actually do something that are in new pathways. But and this is with 2.5 million genomes and 70,000 people. Most of the 28 blood pressure loci they found, where they found signals, harbor multiple genes. So they haven't found any genes, they found SNPs that were correlated that had a whole bunch of genes in the region. And they said, while substantial research is required to identify genes and variants responsible for the associations, so there's no genetic variants, that part of the title is nonsense. Several loci contain highly plausible biological candidates. Well, what does that mean? That means there's no novel pathways because we already know the biology, and that's why we're guessing that those are the right variants, right? So there's no novel pathways, there's no genetic variants. And the lack of significant association reflects small sample size. And this is with 70,000 people, and they found basically nothing that was in the title. The problem is that you spend the next week typing down names of all the co-authors on your paper. That's the list of authors. Letter is 8 pages long, 3 pages of authors, 3 pages of science, 1 page of figures, and 1 page of references. So what I did, I have a lot of friends who send their DNA off to 23andMe and all these companies, and they all think that this is actually real predictive value, and that it tells something about their history or their health. So I thought, well, I'm going to send my DNA to every company that does this and see if they can agree about anything. What I expected was a little bit of variation between groups. I didn't expect the absolute shit show that I got. So this is I'm basically American, which means that anyone who passed through upstate New York, where my family was, in the last 400 years left some DNA in one of my relatives. So I'm as admixed as you can get in America. I'm basically white trash, as we call it in America. And this is my family. It's kind of a bush. There's a lot of inbreeding loops because every small town in the world has them. And my family settled there before the British got there. When one of my ancestors was Dutch. And they were the original settlers of New York, and the British made them take a surname. So everybody with my surname is from upstate New York. This is what 23andMe comes up with, that I'm from all these different places. I'm either British and Irish or Eastern European. Dutch didn't even show up ever. You know, that was the only thing that anyone assumes from my surname. But it's a complete mix of everywhere in Europe. So this might be one answer. But then I sent it to another company, and this company said, this is Ancestry.com. These guys said I was mostly Eastern European and Russian, which I don't know where that came from, because as far as I know, there's none. And then there's like 18% from Europe and Northwestern Europe. This is a secondary reanalysis website that will take your GWAS data and reanalyze it. This one said I have Native American, Indian, Eastern European, North African, and basically everywhere else in the world. So I guess I am the world, right? This is reanalysis of 2 different GWASs, one done by 23andMe and one done by Ancestry.com. And this one here is from 23andMe, said I was 66% Northwestern European and these other percentages. And a different GWAS done by the different company came up with these rather different answers that said I was 21% Eastern European. So this all sounds like similar to each other, right? But then you go to this one. This was sequencing.com that said I was mostly Scandinavian. And then Iberian, like from Spain. They couldn't figure any of this out, right? When I went to this company, they said I was 67% Eastern European and Russian. So it's like, this is all like completely random almost. And I was amazed at how random it turned out to be. But this is where it gets interesting. This company came back to me and said I had ancestors from all these spots with little dots on them. And they said that with 99% certainty, I have a Han Chinese ancestor 4 generations ago. And then they said I had a Punjabi ancestor, and this is statistically determined to be 99% accurate. So I'm really proud. I'm a statistician. I never said anything is 99% accurate in my life. This is even better, though. They did famous people that share my mitochondria. And this one says a mummy in Peru and Eva Longoria. Okay, I'm not sure how that would have happened, but this is the best part. They did my Y chromosome, and they actually sent me this form. This is who shares my Y chromosome, Princess Diana. I didn't even know what to say, except I guess King Charles was hiding something from us. So then I looked at the PRS scores. Now all these companies say they can predict my phenotypes, right? And this is what the general public thinks is the state of the art in science. And they believe it. Because I know people who actually make life decisions saying, I now know something about my health because I sent my DNA away and they told me this. And there's all these different variants. So here's some things that they told me about myself. They said, based on my genetics, I have no back hair and I have no cleft chin, even though I have a cleft chin and a lot of back hair. Then they said that based on my genetics, I'm absolutely more likely to be a morning person than 80% of the population, but I normally wake up at 11. Absolutely not true. Here they said that I'm also very introverted. Isn't that obvious? I'm very shy. And then they said I would have gray hair prematurely. Well, I'm 60, I don't have any gray hair. This is like the crazy stuff that's out there. Then they said, I have a 100% chance of being unlikely to develop a social network. I have 67% chance to be poor. Then I'm gonna have, they predicted my income, okay? And this is what they're sending people and telling them this is genetics. This is personalized medicine in the eyes of the public. Cognitive ability, 100% chance of weak cognitive ability, so maybe I'm wrong about all this stuff. And this is great. They said there's a 100% chance that I'm likely to oppose current foreign policy. Again, genetics, I mean, they don't change every 4 years, and my opinions about foreign policy do. It says a 100% chance that I have no musical ability, but my undergrad was in music, and I'm a professional tuba player when I'm not doing genetics. They said there's a 100% chance that I can't lose weight if I cut fat out of my diet. So I tried and I went from this to this. It was easy. It's just like crazy. And this is what people think is what we're giving them about genetic predictions. Again, unlikely to be good at science. And now this is where it gets good. These are all based on PRS scores about health-related traits. So this one said, now some people say when you look at these polygenic risk scores, only look at the ones that are extreme. Like if you're in the top 5% or the lowest 5%, maybe it's useful. Well, this said I'm in the 100th percentile for insomnia. And sleep is my favorite hobby. I'm very good at it. This one said I'm in the 99th percentile for rapid aging. So I'm prematurely going to be aging and older than I appear to be. I don't think I look that old for 60. But this says I should be accelerated aging. This one says I'm in the 0th percentile for bone density, which means that my bones should be basically rubber, and I shouldn't be able to stand up. But when we were testing our bone density equipment here in Kazakhstan 20 years ago, I ended up being 2 standard deviations above the norm for bone density. So, I mean, these are the things that they're trying to use this for, to predict things for regular people. And you have to understand that this is actually what people think is real science. I mean, I'm the slowest walker in the world. I'm in the 1% lowest range for how fast I can walk. And this is great. This is Alzheimer's disease. So I'm in the 8th percentile of risk, so pretty low risk for Alzheimer's disease. But I'm also in the 8th percentile of risk for resilience to Alzheimer's disease, meaning of not getting it. So it's like, what does this mean? And this is what's out there now. And it's really important to remember that people are taking this and using it. So what basically then is leading is the way we're doing our studies today. So this is the trends in current practice. Ignore what we know about biology and evolution, go on a fishing expedition doing a GWAS to catch something we can imagine is suggestively true, and then do meta-analysis to show it might also possibly perhaps mean something, and then call the New York Times. And we always joke that if your research appears in the New York Times, we know it's crap. Because they only publish the sensational things. What's the European criteria for scientific investment in GWAS? Horrors. The Americans might get there first and take all the profits. What's the American criteria? Damn, the Europeans might get there first and take all the profits. And what's the argument everywhere else? Korea, India, Kazakhstan, everybody else is doing it. Well, the fact is that because everybody else is doing it, especially in a place like Kazakhstan, where you have so many unique natural experiments. You have the nuclear stuff in Semipalatinsk, which you'll be hearing about shortly. You have the Aral Sea situation, which has huge effects on people. And these are really unusual exposures. And then I guess Ainur is doing work in some of the polluted areas of Kazakhstan. These are unique exposures that can give you an opportunity to ask a different question than we ask in the United States. I initially came to Kazakhstan 25 years ago when we were studying the health of the Koreans in Kazakhstan, because we were comparing Koreans who were here, who were sent to the farthest place on earth from the ocean by Stalin in 1937, and we were comparing them with Koreans in Korea and Koreans who were adopted by Swedish families. And the idea was, because of this unique history, you could ask a unique question and see what you can learn from it. So this is what really people should be doing in these other countries, especially when you're building a great resource like you've done here, like Ainur and everybody have built here. Apply it to something where you can actually win. Because if you're asking the same question they're asking in the UK, where they've been doing it for an extra 10 years and have bigger sample sizes, they're going to find it first. But you can ask a unique question they can't ask because of your population resource and because of the unique things that you can study here. And that's really the way to think about it. Don't copy the Americans. Try to do something they can't do because they don't have the resources you have. They have money, but they don't have the population resources, and they don't have all these interesting things that you have. So I often compare science. This is a thing I found once, comparing science and religion. In science, you get an idea, you perform experiments. If the evidence supports the idea, then you got a theory and you use it to better understand the universe. And in religion, you get an idea, you ignore contradicting evidence, you discover new evidence, you ignore contradicting evidence. And that I've changed in my office to human genetics, because we keep doing more and more of these large genome-wide association studies where we don't find anything predictive. We find a whole bunch of loci that do very little. They explain 0.00% of the variance, and they lead to predictions that I'm an introvert with gray hair, no bald spot, and can't lose weight. So I always like to quote Confucius to end with. As he said, it's like learning without thought is labor lost, and thought without learning is perilous. So hopefully that was at least a little bit funny and might motivate you to think about things differently, but to be careful about what you promise to the general public. Make sure that we say things that are based on real facts and not just wishful thinking. Okay.
Thank you very much. Do we have questions from audience for this fantastic, funny genetics?
You're allowed to laugh at me. I laugh at myself. You know, professors are all full of shit, so.
Okay. If no questions, thank you very much.
Yep.
Yeah, that's why we can say that a lot of companies that providing services for genetics need to be very cautious about interpretation. Or maybe everybody will be fat, or everybody will be gray-haired.
Yeah.
Or maybe
but you have to get the bottle that hers came.
Yeah. Or maybe I'm genetically red-haired. Okay.
Yeah.
Thank you very much. Next, our speaker, Vladimir Sayenko. He is associate professor from Department Radiation Molecular Epidemiology, Atomic Bomb Disease Institute, Nagasaki University, School of Medicine, Graduate School of Medical Sciences. And his talk will be about genetic variants and environmental exposure.
My voice strength is good enough to reach everybody. Good morning, everybody. And first of all, let me thank the organizers for the great job of organizing this wonderful convention and for the opportunity to be in front of you today and to have an opportunity to talk. So my title is Contribution of Genetic Variants and Environmental Exposure to Thyroid Cancer. And I feel really grateful to our chairperson and the previous speaker who made a nice introduction for me. He did part of my job, and particularly at the point that when you have unique exposures, you can dissect and dissociate the genetic impact of genetics and impact of the factor you are interested at. So at first, let me remind you about the Chernobyl accident. Oh.
I mean the same.
Okay, thank you. So the Chernobyl accident happened on April 26, 1986, and the Chernobyl nuclear power plant is located at the very north of Ukraine, close to Belarusian borders and somewhat in proximity to the Russian borders. As a result of the accident, a huge amount of radioactivity was released to the environment. Estimates show that the total release was about 13 exabecquerel, and exa stands for 10 to the power of 18. It's a huge amount. Radiologically important isotopes are iodine-131 and cesium-137, and applicably to thyroid cancer, the iodine-131 is the most important. So what happened next? See, the accident took place in 1986, and for the first 4 or 5 years, there has been no change in thyroid cancer incidence, and we call this period of latency, when no disease incidence was changing. But what happened next? Then the increase in the number of cases of childhood thyroid cancer in adolescents and in young adults started to increase. And this pattern is for Belarus. Here on the left is the pattern of thyroid cancer incidence in Ukraine. You see that these temporal patterns are nearly identical. So it gives us an idea that this is a systematic event. So it is reproducible. Well, in 2017, the ANSCIR reported about 20,000 total cases of thyroid cancer, but this was in 2017. Now it's almost a decade later, so you can imagine that the number of cases has increased. Many radiation epidemiology studies have been performed to unambiguously show that thyroid cancer incidence is linked to the thyroid dose of radiation that was absorbed. And what we have learned so far is that there are several risk factors for thyroid cancer after radiation exposure. And so I've listed those here, and today I will be talking about radiation dose to the thyroid, genetic predisposition, and joint effects. Because we have this unique opportunity because of our collaboration with our dear friends from Ukraine, Belarus, and Russia. Although the title of my talk begins with the genetic component, let me begin with the role of radiation dose. Well, almost 15 years ago, we published a GWAS study, which was the second in the world GWAS study, and first in the world that was analyzing radiation-related thyroid cancer, and it still remains the only one. At the time of the GWAS study that I just showed you, we did not have information on the individual doses, radiation doses to the thyroid. And this was the major limitation of the study. Therefore, we made some kind of an effort, and it was a great international collaboration in which our colleagues from Japan, Belarus, the United States, behaves itself, the United States and France, Jesus. We made a team and reconstructed individual doses together. So let me briefly explain what we had in our hands. So there were cases, about 1,000 participants, whose age at the time of the Chernobyl accident was below 18 years old. This is a risk group. They were residing in radio-contaminated areas, and they all had histologically verified diagnosis of the papillary thyroid carcinoma. Inclusion criteria for controls are almost the same that for thyroid cancer cases, but they were euthyroid state, and of course they were the same age at the time of exposure and had no thyroid abnormalities on ultrasound screening. This is distribution of our study participants. The participants were recruited from Belarus, which is a heavily contaminated country. When the dosimetry team performs their dosimetric investigation, they should be blind toward the knowledge who is a case and who is a control. So they perform their modeling and dose reconstruction in a blind manner, and they gave us this result. But after that, and after we break the codes, we can plot the dose distribution for cases and controls. Cases are shown in pink. And as you can see, the doses in cancer patients are higher than in the controls. So we can, from this, we can get an idea that there is some role of radiation indeed. One of important points highlighted over there in green is that our groups of cases and controls were well balanced for sex and age. These are baseline characteristics, and dose accumulation to the thyroid strongly depends especially on age. I'll show you a bit about that. And what we do at first, and we just check the association of sex, age, and exposure, and thyroid dose, of course, with the outcome. And as we can see, there is no impact of age. And what is interesting from sex, and what is interesting about the age at exposure, that it gives us an odds ratio less than 1, although it does not fit 95 confidence interval, but with the CI of 90%, we can see that the older the age is, the lower the chance of developing cancer is. However, if we analyze the group that was exposed at the age below than 10 years, we can see very nice association. So it means the younger the age is, the higher the chance to develop thyroid cancer after exposure. We can observe, we can expect. And then we can split all thyroid dose data into quartiles and estimate the effect of thyroid dose for each quartile. And here we can see the increasing effect. The higher the dose is, the higher the chance of getting thyroid cancer after exposure is. So it is rising. And what is important, I didn't mention, that if we consider only thyroid dose in a statistical model, we can observe that it sort of explains about 13% of variance, variance was several or many times mentioned in the previous talk. So it is 13% only, but not 0, not 1. Although it's not a population study, this is a case control study. Now let me move on to genetic predisposition. And again, this slide was already shown. That study was performed about 15 years ago, and since that time, many other GWAS studies were performed, but they were all, once again, performed in principally non-irradiated, non-exposed population case controls. So many other SNPs came out to be significant, and we decided to focus on 6 of them located to 4 genetic foci that appeared to be the strongest worldwide. We've performed many other whole genome sequencing and chip SNP analysis. So we increased our group, and then since we use several different platforms, we extracted data for these 6 SNPs and analyzed those. Just a bit explanation about logistic regressions. If we consider the effect of the genetic component of SNP, we put just one SNP into the model, and we judge about the chance of outcome. If we add sex and age to the model, we call it sex and age adjusted effect of SNP. And if we put sex, age, and many SNPs, we call it multi-SNP model, sex and age adjusted. And what is important here is that we can evaluate the effects of each SNP that we've analyzed, non-adjusted and adjusted. And it is necessary to say that when we analyze SNP one by one, we got 4 SNPs that showed the association signal. In the multi-SNP model, only 3 survive. And if we combine them in one model, we can see that they all 3 remain statistically significant, and they add about 5% to explanation of variance. Then, so we just heard about radiation, about genetic effects, and now let's think whether they have combination effect, or we call it joint effect. And so what we do is we put into the model a SNP effect, a dose effect, and then SNP and dose effect. And at first, what we see here, that all 3 SNPs remain statistically significant here. The dose is, of course, statistically significant. And altogether, they explain about 18% of variance of outcome. And what is important also is that negligible change in the effect size in terms of odds ratio tells us that SNPs and radiation contribute to the outcome chance independently. I can often see in the papers that the authors mention when they perform a multivariable model that those variables that remain significant after combining them into one model, they call it independent effect. No, it's not an independent effect, and if there will be questions, I would be glad to work out on that. Joint effect. We have radiation effect, we have genetic effect. So now what's going on, what happens if we estimate their joint effect? How do we do that? If we supplement our model with the so-called interaction term, highlighted here in yellow. And for one SNP that was significant, that had significant association signal, appeared to yield a significant interaction effect. And what is important here is that, as you can see, odds ratio for this interaction is less than 1. And this has very important implication. We now can plot this interaction effect on these two slides, the effect of genotype by the dose and the effect of the dose by genotype. And because this is very important, let me enlarge this graph on the left. See here, thyroid dose is plotted on the horizontal axis. And when we check what the impact of the genetic component is, we see that it declines, which would be expected from the odds ratio below 1. But what's important, that somewhere it hits the odds ratio of 1. And this happens somewhere here at the dose about 120 millisievert. So after the higher dose, we don't see the effect of the genetic component anymore. So at low doses, there is contribution of genetics. But after some, well, I wouldn't call it threshold, but if you wish, let it be a threshold. For this given SNP, we do not see genetic contribution to the risk of thyroid cancer anymore if the dose exceeds 120 millisieverts. In contrast, whatever number of risk alleles, 0, 1, or 2 we have, the effect of the dose remains statistically significant. So the dose sort of overweights the effect of the genetic component. Let me skip it. Okay, finally, we combine everything into one model, and we can see that our model that includes 3 genetic components, 3 SNPs, the individual thyroid dose, and their interaction term, it explains 18 to 19 of variance of outcome. And that's basically it. I have some conclusions over here, but let me not to repeat those. But in the final words of my presentation, I would like to leave you with an encouraging message shown in blue at the bottom of the slides, that we do not have good, strong evidence that genetic component and radiation dose enhance each other effects. Philosophically, let me summarize it as so far, there is no evidence that there are people genetically, specifically predisposed to the development of thyroid cancer after radiation exposure. And that would be my take-home message. I would like to thank our great friends and collaborators from different countries all over the world. And from Belarus, we had a wonderful team whom we are getting in touch so far, and also would like to thank Dr. Svetlana Mankovskaya, who is attending this meeting. And here I would like to stop thanking you all for your kind attention. This is a must-see place in Nagasaki, which is called Spectacle Bridge. Thank you.
Does anyone have questions? Yeah?
Mutation in BRCA would predispose to cancer?
Not in thyroid cancer. It has a little effect on the level of SNP. As a kind of, well, by definition, thyroid and ovarian cancer.
Cannot exclude that some genes would predispose strongly.
Well, the phenomenon of missing heritability is still here.
it's okay.
So yes, but its effect might be very, very small. But yes, you're right.
I mean, I was, when you did the analysis of interaction, basically you were starting with SNPs that were identified in thyroid cancer with no radiation, right?
Yes. Yes.
So you're starting with ones that you know have an effect when there's no radiation. And you know radiation has effect when there's no genes. So it seems almost natural that they would appear to have that kind of the negative interaction, because the radiation itself is such a high risk factor that the genes are no longer needed and they're basically irrelevant. That's kind of what it's saying, right?
Yeah, yeah.
If you're really, like if we drop a bomb here, you're going to be in trouble whether or not you have the right
yes,
genes,
yes,
right? And the genes aren't going to affect that. That's basically your conclusion, right?
Yeah, that's basically. And also my conclusion would be, you're right, that contribution or impact of radiation exposure, which explains, let's say, 13 to 14% of variants compared to 4 to 5% of genetic components, is almost 3 times.
Right, exactly,
Right, times. Also, I would like to mention that here we analyze SNPs only, but since we have GWAS data, there may be that other types of genetic variations might be discovered.
Right, that's what I was getting to, is like, because you started with SNPs that were identified in an unexposed group, and you're looking at an exposed group. In order to find anything with positive interaction, you'd be looking for GWAS hits that didn't appear in the unexposed group. So it's natural that I think that the ones found in the unexposed group would have less effect in the presence of radiation, because the radiation is so overwhelming, especially the high radiation, which is where it was the most extreme, because that had an even bigger effect than you were saying. Yeah.
Oh,
yeah. So it makes sense. Yeah.
Well, good that at least somebody finds that makes sense.
Thank you for the presentation. Maybe ask stupid question, because I'm not specialist. My question is, how someone define the radiation dose after 20 years of exposure, or how many years left after the exposure, right? So how to define it? Yeah, just
although
I'm curious.
Although I was the last author on the dosimetric paper that we've published describing these findings, well, dosimetrists, dosimetric team members, they use very sophisticated models to reconstruct the dose. Also, we had collaborators from IARC in France, in NIH, and by chance, occasionally, we discovered about 60 participants, 60 cases that were participants of previous studies, which had direct thyroid measurements. And then we had subgroups of patients for whom we had very good description of their information on their geographical location, of course their sex and age and exposure. So by making some kind of empirical models, dosimetrists can determine the dose on the individual basis, but they are using different models.
Thank you.
Thank you once again.
Sorry? There's a question? Yes?
Wait.
There's one more
It's kind
question.
of too late.
One more question.
Just a quick question, I'm just curious.
Sorry.
Sorry, So you mentioned in the beginning of your talk, you showed those two graphs with incidence of cancer, and I think in Russia or Ukraine. So you showed
In Belarus and Ukraine.
yeah. And there was like latent period
Yes.
from 1986. Do you know what this could be related to? Why? How can you explain that? That latency, that whole 5 years of.
Well, radiation-related diseases, whatever it is, cancer or non-cancer disease, is a stochastic event. So it occurs by chance. And if we consider that radiation was the initiator, so it started the disease, it takes time that disease reaches its detectable stage. So it takes time. None of the diseases occurs immediately, right? I mean, like cancer.
I guess there's one more.
Two more.
Two more, okay.
I'm happy we have this live discussion, though.
It's an interesting talk. You're going to get questions.
Okay. How to protect thyroid if there is expected or really occurring radioiodine release? Well, our radiological protection logic suggests the ALARA principle. So, shelter, restrict time of contacting with radiation, take protective measures. So in case of radioiodine, there's a good approach. Take stable iodine pill. It blocks the thyroid. It is entrapped to the thyroid and blocks additional entrapping of radioiodine that was ingested. So that's the solution, and that's why potassium iodine pills are distributed in some countries according to local regulations to the population, to residents who live nearby in the vicinity of nuclear power stations.
Okay, thank you. Yeah, okay, we have to move on. So the next speaker is my good friend Ainur Akilzhanova, and she's going to talk about the genomic studies she's been doing here at the Center for Life Sciences and how it's paving the way for personalized medicine in Kazakhstan, all the great work she's been doing for the last 15 years.
Thank you. Thank you. And previous talk, it was many questions, and I think it will be also a good chance for us for collaboration enlargement, because in Kazakhstan launch that will be nuclear power plant opening maybe in few years later. That's why this radiation research is very important. Also, we had this power plant in Semipalatinsk region. Still, we have many scientific work on this direction. That's why it's very many questions arose, Vladimir, your talk. But my talk regarding genomic studies which were done during these 15 years, and of course we are not in the personalized medicine era yet, but still we are approaching here very quickly, and hopefully with the help of our collaborators in the interpretation of data, in analysis of data, and functional genomics, we can further advance this area. And I will do just overview, because of time not so many for me as an organizer. That will be short presentation. Please, next. I will do myself. Okay. As we know that we differ between each other only by 0.1%, but this 0.1%, it will be enough to study different predisposition to conditions, different diseases, etc. And here you can see only example of genetic card of the first chromosome, and we can find different losses that lead to different diseases. And genetic variation, of course, can be given inherited from parents to offsprings, and determine specific traits and specific disease which can be studied and can be analyzed by further studies. In Kazakhstan, we established laboratory from 2011 for genetic research, and of course our instrument was in that time platform Illumina HiSeq 2000, but nowadays we have Illumina 6000, and also from 2020 we have Oxford Nanopore platforms, and next year, hopefully, if our collaborators from Illumina will give us discount, we can buy further platforms to increase our capacity for sequencing facilities. And we can use not only whole genome sequencing, but also hotspot panels, actionable panels for disease-focused panels, and comprehensive gene panels, including whole exome sequencing to establish this foundation for personalized medicine in Kazakhstan. And this also becomes true, and we included in our scientific direction different genomic studies, including not only in multifactorial disease predisposition studies, but also in mutation analysis in biodiversity of Mycobacterium tuberculosis, of different disease-causing factors, also applied genetics in agriculture, applied genetics in wheat genetics, and horse animal genetics in Kazakhstan. And recently, we also study and switch our projects in the field of functional genomics, because we found several novel mutations which can be causing factors for different conditions, and we need to validate if these novel mutations lead to this disease or not by using iPS-derived technologies, CRISPR-Cas technologies, and so on. And thank you for our collaboration with Dr. Bart in this field. And here just overview and small numbers of samples which we already sequenced by using our different platforms and combination of these platforms. And of course, core of our genomic study is a Kazakh architecture project, and during this project we're going to different regions of Kazakhstan, and just recently we went to Aktau. This is a West Kazakhstan region where we was for the first time, and we want to include in our geography and regions of Kazakhstan every part of this population, of Kazakhstani population, to this Kazakhstani genomic map. And we already include Central Kazakhstan, East Kazakhstan, West Kazakhstan, and this around 1,000 genomes will be core of Kazakhstani reference database. And meanwhile of doing these architectural populational studies, we're doing clinical genomic part. In these projects, we collaborate with different hospitals, and one of them is a Center for Heart, National Cardiac Surgery Center, collaborating from 2010, and we have several projects together with them, studying different cardiovascular disorders, including arrhythmias, channelopathies, atherosclerosis, LVAD, left ventricular assist device persons, to establish this genomic usage of these methods for their diagnosis and for their treatment. Also, we continue with this year with epilepsy, continue with using genetic technology to our athletes in national Olympic teams. Then we also study neurodegenerative disorders, which will be in the session of Shulpan. She will show data. And of course, we have several genome data of horses, and hopefully tomorrow or today we will have a chance to eat horse meat, and we study why our horse meat is so tasty or not, because our horse species is very resistant to heavy winter, heavy snow, and freezing, and we study from genomic part. And also we continue in collaboration with Academy of Sciences and Dr. Torospekov about wheat genomics also, and of course, we also did a genomic study of the Cervus elephus. Is antlers, which will be very good and say is a prolonged life. We have very good farms in the East Kazakhstan where these antlers producing, and many treatment sanatoriums for that. If we say about cardiovascular disorders, and this is the main point of my scientific interest, because I am cardiology by training, and during these projects we trained our young scientists, and of course main part is bioinformatics pipelines and bioinformatics facility. Later Ulukbek, he will give us information regarding Kazakh genome and about bioinformatic pipelines which we are using. During these 15 years, we established from the scratch, and that's why I can say that now we can analyze maybe 100, 200 genomes very quickly and simultaneously. And hopefully, if we'll do more collaboration, we can analyze thousands of genomes and expand our capacity for 10,000, 20,000 of Kazakhstani population. If we say about cardiovascular disease, it's still on the first place among mortality and morbidity, and many complications like myocardial infarction, insult, stroke can lead to death, and that's why very important to study at least 30% of genetic predisposing factors to these cardiovascular disorders. And since 1990s in US already established that genetic tests will be very useful for diagnosis, for prognosis, and for prophylaxis in cardiomyopathies, heart rhythm disorders, vascular disorders, and lipid disorders and hypercholesterolemia. And during our projects, we study not only atherosclerosis, but also channelopathies, arrhythmias, rhythm disturbances, and you can see even in only one atherosclerotic condition, so many genes involved in predisposing factors to this disease. And we have two big programs together with the National Cardiac Surgery Center, where we study genetic basis of atherosclerosis to establish risks for this condition. And also we had one program, it's a 350 whole genomes was sequenced in frame of projects of post-COVID syndrome, where cardiovascular complication was the main conditions for these COVID patients. And one of our also project and direction, genetic of channelopathies and arrhythmia, where we established cardiogenetic panel. It's a targeted panel including 96 genes involved in cardiomyopathy, different types, and different types of channelopathy. Using this panel, we validated on different ventricular tachyarrhythmias, atrial fibrillation, and Brugada syndrome, and long QT syndrome, and etc. And it was give us chance to give direct and correct diagnosis to these patients. Of course, hereditary of cardiomyopathy very complex, and we also established this panel and exome sequencing to do correct diagnosis in different types of cardiomyopathy. And if, when we found some novel mutation, now we're further establishing this functional genomics for same patient to see this etiological value of this novel mutation in our patients. And this panel, which include 96 genes, of course, it's based on existing panels, but we included another genes which was involved and associated with these diseases. Also, we using further these genetic panels and exome sequencing for molecular autopsy. We have also collaboration with the Astana Forensic Center, where we see patients who died under age of 40 because of sudden cardiac death. But what we found in no, this is different. Yes, in sudden cardiac death, we found two groups who died in age younger than 40 because of myocardial infarction, but they don't know about their disease, about their diagnosis. And second group is the secondary cardiomyopathy, where no any diagnostic changes found in heart, but they could be die because of asystole or because of fibrillation of the heart due to mutations in the ion channels. And we found that 63% of mutation was found in group of non-diagnostic structural changes, and 35 mutation in the genes was found in cardiomyopathy post-mortem. It means who died due to myocardial infarction without knowing this disease. This shows that our younger population don't know or maybe not pay attention on their disease and their symptoms, and they don't going to hospitals to be diagnosed, and they die because of sudden cardiac death because of these conditions. But second group, maybe we can increase our capacity to sequence and found the genetic causes of their condition. This mutation which we found also. Another group, LVAT. Today in the morning was one report about transplantation of the heart, and this program about transplantation was begin from 2010 in the heart center, and already we have transplanted a lot of hearts and LVATs. And in this patient, first 100 patient, we did genetic analysis for genes which involved in thrombosis and involved in the mechanism of antiplatelet drugs and antithrombotic drugs, because such patients need to be anticoagulant treatment and antiaggregant treatment whole life. And genome-guided assessment give us chance to give direct dose for such patients. These just examples where we can use this genetic test, and cardiogenomics enables better patient outcomes, and this is possible to do at any age. If we will begin from the young age, it will be better chance to provide prophylactic measures, but if we will do during disease, we can do secondary prophylaxis, but still we can find solution for right treatment through pharmacogenetic test during these diseases. That's why when we're combining different genomics, proteomics, and metabolomics, this gives a chance to give a better personalized treatment and care for our patients. And if we will find pathogenic variant, we can confirm diagnosis, we can guide treatment, we can monitor response to the treatment, and we can test cascade testing for relatives. If we will find benign variant, we just follow standard treatment and protocols of Ministry of Healthcare. And if we will find variant of unknown significance or novel mutation, we can lead further for our functional genomic studies. These are just cases showing that our results can be useful for our clinicians. And of course, using IPS cardiomyocytes, we can do experimental model of cardiomyopathy or experimental model of cardiomyocytes on Petri dish, and then further study pathophysiology of these diseases, or study maybe drug action on these cardiomyocytes. But this, of course, we just begin. We are just babies on this, but hopefully in future I can present results of this, our part of the studies. Thank you very much. And of course, this data about Kazakhstani architecture will be later show results, because we are very different and we are very heterogeneous, and it's also very challenging to study Kazakh population, but also very interesting to study Kazakh population. And you can see Khazars, Kazakh, Kyrgyz, we are situating in the Central Asia, and we are sharing this heterogeneity between this population. And our genomics might be mapping as a geographical map. And our team is already not young, already 15 years old, older, and still we have our laboratory members, and we have laboratory for bioinformaticians, and this our previous conference photo where we had in 2022 or 2020, where we had 10 years of our Center for Life Sciences. But still, gradually we can improve our capacity and improve our scientific abilities. Thank you very much, and thank you very much for our collaborators and sponsors who give us chance to increase our capacity to do science. Thank you.
Are there any questions for her? I mean, I noticed that on your slide where you showed the genetic where Kazakhs fit, that it's almost fitting the joke I used to hear that when a Russian and a Chinese have a kid, it comes out Kazakh.
If 3 Kazakh persons will gather together, 2 of them will be relatives.
Yeah, exactly.
Yeah.
Any questions? I mean, that was a lot of stuff she talked about, so I think that we have to digest it all because she's done so much work in 15 years.
Thank you.
Let's see. So the next speaker is Dr. Richard Carter, going to talk about data considerations in biotechnology.
Yeah, Richard Carter, he's supposed to be here, but today, Dr. Bhupinder, he coming together, and he is a founder of the company, and he will present instead of him.
Some of you must have wondered, he doesn't look like Richard Carter. Thank you, organization committee, especially Professor Doss and Professor Ainur, for making this possible for us. to present here. My name is Bhupinder Handal, and I'm representing on behalf of Richard Carter, who couldn't make it today. Richard Carter and I worked over 10 years with Oxford Nanopore as senior directors, and this year we decided to form our own company to provide data management services to the scientific community. So in this slide, I present that in order to build a biotech data project, it's like building with Lego, and each color represents the Lego. Different Lego pieces represent various considerations that you need to pay attention to in order to do experiment. So it's sample, experimental, infrastructure, and data analysis considerations. Within each of these considerations, you have to have deep dive, and you can put these pieces together in various orders. It doesn't have to be that way, so you can customize it for yourself. So let's start with the first one. So for building a project, you need a sample source. Sample source for NGS projects is very important because sample could be a blood sample, it could be frozen, could be solid tumor. Sample extraction method, what extraction method are you going to use? Consent for analysis, if you're dealing with human data, the consent is very important. Library preparation method, that's mainly determined by what type of technology you use. If you're using Illumina, you don't have to worry too much about it because you're doing PCR. But for nanopore, for example, you are dealing with long read, and you're actually measuring the DNA going through a pore, so you have to worry about contamination. You have to make sure that the pieces are, they're not broken up into small pieces. And the sample tracking, how are you going to track your samples? Is it automated process or not? So a number of samples. If you're doing 10 samples, it's easy to do. You can record in your book. You might need automation. Experimental type, what type of experiment it is. Is it a whole genome project or a targeted genome? On-device monitoring, do you need on-device monitoring or is it done at the end? For example, in Nanopore, you can do monitoring at the same time as you're running the experiment. And QC consideration. QC must be done early on to make sure that you're not wasting your time and resource. So that make sure that you're going to get your answer at the end. Data handling. How are you handling your data? Are you transferring to a server or are you storing on your device? So data transfer is very important. You need a right type of network. Temporary storage. So you make sure you have enough storage while you're in the experiments. If you run out of storage, you waste your experiment. Compute. Compute is very important because you might be analyzing your data in real time or afterwards. So if you're using, say, 50 CPUs, is that enough for you? It might take too long. You might need to have 10 times the compute, right? And if you use 10 times 50 CPUs, that'd be very expensive to run. Disk space, ensure that you have enough disk space. Do you want to archive your data? Is it important? Is it important to save 2 years or 3 years? What would you do with it? All that is very important. What kind of analysis workflow are you using? Data formats, what kind of data you have? Is it a FAST5 or FASTQ? Analysis QC, make sure that you do QC early on and you are doing the right type of analysis so that make sure that you're getting end result the right. Continuing analysis reports, make sure that you're doing analysis all the time in order to make sure that the experimental integrity is maintained so that you're getting the right result. Data sharing permissions. Don't allow especially confidential data to be shared with everybody. There are other considerations in doing such projects. Automation will help for data transfer, analysis, and metrics gathering. Freezing software versions. because sometimes the new softwares keep coming. So you want to make sure that you keep the software consistency, lock it. Otherwise, you will not be able to compare the data from one sample to another. Don't share what you don't need to. You need to be aware that you're handling human data. It's very restricted, and it's very confidential. Measure everything. Report what's important. You can use dashboards or sample clinics to make sure that the wet labs are getting all the feedback that everything is working smoothly. So hence, experimental feedback is important. In order to establish rapid turnaround times, again, provide the feedback. Also remember, experiment success is not just dependent on running the actual experiment, but sample handling. Who is doing this? When and how? So you want to track each sample, make sure who did it, and if they made a mistake, you will be able to figure it out. Automation, temperature and humidity. This is very critical sometimes because some machines won't work at certain temperature. You can't have nanopore running at the same time as Illumina in the same lab. Make sure one doesn't heat up the other. Oh. So we see at Legion, we see that there are 4 stages of doing an experiment. Proof of concept, proof of value, proof of scale, and production. You must run a proof of concept. If you're running a 1,000 genomes project, make sure you've done 10 or 50 samples and see that you're getting the end result. And in order to run a large experiment, then you want to do proof of value. So you want to upscale. You want to see that, are you getting the right value? If it's too expensive to run, you don't want to do this. You want to make sure your cost is worthwhile doing the work. Proof of scale, are you able to scale it up? Because you might be able to do 100 samples, but if you want to do 1,000, you make sure that you have capacity. And then you want to take to the production scales. So Rich and I worked with lots of projects, large-scale projects. For example, at the Emirati Genome Project did 200,000 genomes, and we installed nanopore over there, and we managed the whole project. We did the Singapore Genome Project. We did the Dubai Genome Project. And we can help our experience to take you from proof of concept to all the way to the production stage. This is what we will provide. We can also use various tools for the fast adoption by using new techniques. So AI is now used a lot in doing the analysis. These are various platforms you might be aware of. So one can use these AI tools to create the code. You can simply write a script to align a sequence. It will give you a code, but it won't be fit for purpose. It's like providing scaffolding. It gives you can use this for proof of concept, but you can't use it for clinical analysis. Left unchecked, AI code can leave a product far more buggy than what humans would produce. This is an article came out 2 days ago in TechCrunch. It says, using AI is like hiring your stubborn, insolent teenager to help you do something. You have to ask them 15 times to do something. In the end, they do some of what you asked, some stuff you didn't ask for, and they break a bunch of things along the way. Sorry, it's very hard to read at the same time. Is that next slide? This is. Okay. Am I going forward or backward? Going backwards. Yeah, okay. So one can run their own AI solution and train different models. There are some examples at the top. This is called retrieval. You can give your own sequence into the system and use these platforms provided by AWS or Google or Copilot. These are big platforms one can use. There are also bespoke models you can use, bioinformatics models. These are custom designed by the private companies, but they can be very expensive, and they require expensive servers. There are also BioTrack-trained models available, which are highlighted there. And on the right side, we have various variant calling programs, which are either commercially available or they are open source provided. So one must consider data security very seriously. You're handling human data, and it's very important to keep it confidential to make sure that you are meeting GDPR requirements and geographical restrictions are respected. The commercially available cloud platforms such as AWS and Azure, they claim to provide you security, but make sure that you have limited access to the permissions to the people. At the end, customer is responsible for the compliance part. Don't store your data if you don't need to, because it can be very costly. Provide least privileges to people if they don't need it. Careful where you share your data, especially on AI platform. Once you put your sequence in an AI platform, it will be available to everybody. So at Legion Bioscience, we will provide you global expertise from our own experience. Unbiased advice, whether you use short read, long read, whatever platform you use, we can help you with. My colleagues have been working with Illumina, with Solexa, with Nanopore. We provide you practical results, hands-on results, and we have no vendor preferences. We can also help you with proteomics. Proteomics is much more complex because of the protein structure, of course. And the data can be 2 times more volume and difficult to handle. So we have expertise both in genomics and proteomics. So please come and talk to us after the talk. We'd be glad to talk to you. Thank you so much.
Thank you very much. Anybody have questions? Yeah, good chance to have a good partner for collaboration for further analysis of your data on genomics, proteomics.
Thank you.
Okay, thank you. Thank you very much, Vipinder.
Okay, next speaker is Kairov. Going to talk about towards to understanding the landscape of Kazakh genomes, insights from multi-technological approaches.
Okay, good afternoon, everyone. So I'm happy to welcome you on our conference. And today I would like to share some insights about the Kazakh genetics. And so we will talk about the recent discoveries. Most of the results are still unpublished in the preparation, but some of them are already there. So as you may know, the Kazakhstan is situated alongside the Great Silk Way, and it's habited mostly by the nomadic tribes, and with the majority of it's multi-ethnic country with the majority of the Kazakh ethnic group. And if you would like to see this, how many genomic studies was done on the Central Asian region, so there are still there are some very big limitations in that case, and there is only few papers are published related to Kazakhs, and there is very limited number of papers in English related to other nations of Central Asian regions like Uzbeks, Kyrgyz, etc. And some of the papers started to be appeared in the scientific literature starting from 2020. And as you can see here, the first paper was published by Korean group. It was related only about the one female sample sequenced on the NGS platform. Then half of the papers was published by our center and our groups. So one of them about the data description of the sequencing of first initial samples of Kazakh whole genomes, and some of them about the insights of the genetics architecture and genetic variants. And the last paper was published recently by Chinese group. Chinese group about the Kazakhs residing in the Xinjiang region of China. This is the brief overview of the sampling collection map that we use. So that we collected all the samples during our expeditions, and in terms of collaboration with different clinics from different regions of Kazakhstan. And as you may see, that's, the sampling collection is not still uniformly distributed, but we are still trying to continue our expedition in different regions. And just recently, we've been visited this west region of Kazakhstan, Aktau region, Mangostau region, so we're collected and we're planning to sequence them and to look deeply in the genetics of the people who's living in those regions. So today I would like to talk about the multi-technological approach that we used to decipher, to investigate the Kazakh genomics. So, and some of the slides will be related to different types of data that we are investigated. And so we have collected data from 500 samples generated using the next generation sequencing platform. Then we have 500 samples. So I'm talking about the only relatively healthy people and healthy Kazakhs, but also, as Professor Akilzhanova mentioned, so we have in parallel several different projects related to the clinics, so we are still collecting and trying to sequence also clinical samples. And then we have around 500 samples generated on third generation sequencing platform, Oxford Nanopore 48. And then, a few years ago, so we are genotyped using microarray technology, so Illumina GSA24 platform. And so, of course, this generation sequencing itself, it's the first initial step, and then there's the more critical steps there, it's analysis part. And during the last year, so we were able to implement different types of pipelines and bioinformatics processing algorithms, etc., in order to make all future results effective. And of course, in parallel, we are trying to develop our clusters and computational facilities, because it's a very important step also. Just you have to storage, manage, and analyze the big amount of genomics data. So this is the brief overview. I'm not going to go deeply about the quality assessment, etc. So I will show you some different slides because of this time limits. I will show the most important slides here. So this is the distribution of different genetic variants that were identified in whole genome sequencing data of Kazakh population. As you can see here, the most of the variants are located in non-coding genomic parts of our genome, intergenic and intronic, but we still have some interesting variants which are identified in exonic parts also. And most of the variants by functional type, the synonymous, thank you, synonymous and frame shift variants. And here the key number here is, so we are trying to calculate the average number of SNPs per sample, and you can see the number here is around 5 million SNPs per one sample. That's the average number, but it's this number is including both types, novel and known SNPs. So this is the last paper related to final third phase of the 1000 Genome Project. It was published in 2015, and you can see here there's the average number of SNPs per different populations here. And as you can see here, the European population has less number of SNPs per individual, and South American population and African population has greater number. So the Kazakhs among them, then very close to African and between this South American populations on the level of the SNPs per one individual. And then we try to map the whole genome data that we have in comparison with different populations. So we collected the data from different data sets, global genomic projects like 1000 Genome Project, AGTP, Estonian biobanking data sets, etc. And you can see here, so we collected the Eurasian populations and tried to map our Kazakh whole genome data, autosomal data on the geographical map. And the key message here, so that's that whole genome, so whole genomic information fits geography very well. So this is the results from whole genome data. So based on the more than 230 samples. And you can see here the same, we repeated the same analysis for GWAS data, so genotyping data. So that's also around more than 220 Kazakh samples. And the same pictures, same distribution, so that if you give me your whole genome, I can somehow identify your place of origin, so, and find your closest population. So I see here that East Asians are always on the right side. So Siberian, Eurasian populations close to Central Asian cluster. Then we tried to look at the whole exome sequencing. So I mentioned before, so we use 3 types of data sets generated by different genomics platforms. And the same distribution, so that's related to whole exome data, so on the exonic part was sequenced and analyzed. So the same distribution, so, but this is, these samples are combined with the data from 1000 Genome Project, so that's the population. Composition is completely different comparing to the Estonian Biobank data sets. And then we try to make an assessment, so by using specific tests, how many, so using the K3 test, etc. So how Kazakh genome are mixed, so what main parts of what mixture are existing in Kazakh genomes. So we can see the different genetic workflows, mostly coming from East Asian and European population signals. Then this is an example of our published paper just recently about that mixture. So we see here the clear signal from Central Asian region that Central Asian populations are very highly admixed. Then we try to make an assessment of fixation index. So fixation index usually very often used in population genomic studies to make an assessment of the close relative populations in, for example, in comparison with your samples. So when you can see here the less number is much closer and greater number. This is the heat map on fixation index distribution. And so if you look, so this is the very dense figure, but if I would say that top 7 population closest to the Kazakh population, so the first one will be Karakalpaks, then Kyrgyz, Uyghurs, then Hazara, Kalmyk, and Altai. It's very interesting. The Hazarian people, they don't have their own stateholds. but they distributed in the Central Asian region in different parts. In Afghanistan, some of them are residing in Turkey, Iran, etc., etc., but they all have the Turkish origin. It's clearly seen here. They're very close. So the Hazara, Kalmyks, and Altai people have the same numbers of fixation indexes. That's why they're depicted here after the slash. Then Uzbeks, Buryats, and Bashkirs are closest populations, based on the whole genomic level. Then I'm not going to stop on the very interesting list of SNPs and insertion deletions, and I will stop on the most interesting, connected with phenotypes and some diseases. So as you can see, the distribution, minor level frequency, in comparing to European and East Asian populations. So you see here there are some increased risk in celiac disease in our population, but much less than comparing to European populations. So there are some SNPs connected to melanin pigmentations, and if after the comparison with clinically significant variants using this CleanVar database, we found that very interesting SNPs connected with the pharmacogenomics, though there is some good examples, for example, related to atorvastatin response and hypertension treatment also. Then this is the very popular SNPs which are assessing using this whole genome data. So first one is lactose tolerance, and you see here, so we have less number of predisposition comparing to European populations, but higher than comparing with East Asian populations. And there are some two interesting genes here involved in the oxidation of ethanol in our body. It's alcohol dehydrogenase and aldehyde dehydrogenase. So there are two genes are encoding the enzymes which can help us to make an oxidation of ethanol in our body. So if you look at this distribution and frequency here, they're statistically very significant. So if you look here, so we are much closer to European population than to Asian. So there is a very known symptom called Asian flush. So connected to this Asian flush, it's related to the fast and slow oxidation of the ethanol to acetaldehyde. So we are closer to Europeans than to Asians. Then we try to make an assessment of inbreeding level in our population, and the key message here, so this is the figure, you can see here that Kazakh has the lowest number in breeding coefficients. It's measured by runs of homozygosity. And as you know, the Kazakh nation has cultural and traditional norms strongly prohibited to consanguineous marriages. So that's clearly seen here. That's how the cultural traditions can be reflected on genomic level and vice versa. So that's, I mean, this good knowledge of good knowledge of genealogy up to 7 generations can give us some good effects. So we have reduced risk of recessive genetic disorders. Then this is the last result, so we tried to make an assessment of the integration of the ancient human genomes. This is the very known fact that European population has a bigger fraction of Neanderthal integration, but the Asian population bigger fraction of Denisovan Asian genome integration. So this is the Denisovan and Neanderthalian genomes, the ancient genomes of hominids, which are disappeared, but we still see here. So we are compared only with Denisovan. This is the last data. So, and then in the future we will show the results of comparison with Neanderthal genome. But using this Abba test, we calculated that this statistics has positive value. It means that we have the bigger fraction of Denisovan integration comparing to Central European ancestry samples. So we used, as an outgroup, we used African population, Yoruban tribe from Africa. Then at the end, we also tried to calculate the possible coalescent times in last admixture events that was happened in our population based on the analysis of more than 200 whole genome data. And the key message here, so that's we calculated the possible time of how many generations ago the last biggest admixture, even on genetic level, was has been in our population. And this, we calculated it's over 24.5. But if you calculate, it's not just a number, so if you try to calculate, if you look, that's the average length of human generation, it's around 25 years, and if you multiply, you will get 562 years ago. It's mid of the 15th century, and it's very close to the beginning of formation of Kazakh statehood. It was organized and started by Janibek Khans, and so the initial steps of establishment of Kazakh Khanate. Then, so we tried to use long-read sequencing data in order to make our steps towards creation of Kazakh reference genome. And there's the last results of the assembly of three individuals here. So we used hybrid approach here, so the good quality assessment by Busko here. And then we are collected data from the already published in our assembled genomes from East Asian populations, and our numbers are close, similar. So in some of the cases, especially in case of number of objects, we have better numbers.
Лукбек, время.
Yes, okay, so I'm finished. And then at the end, I would like to announce, and very happy, that we are launched our Kazakh genomic variation database recently. So, and it's work in the pilot mode, and will be available to all of the researchers to see the frequency of distribution, different genetic variants within the 1 month. And there's very useful database for biomedical research and clinicians. So there is a main page of interfaces you can choose, or you can input any genome position or RSID in this database and look at the frequency. And there are some information will be included related to different populations also. So so the final last slide about the future plans, where we're going. The first one, of course, we would like to expand our genomic database with different data sets coming from long sequencing data and genotyping data. Then, of course, we would like to improve our de novo assembly going forward to creation better reference genome of Kazakh population using high-C technology and optical mapping. And then the final goal, so we have to create a Kazakh pan-genome panel to better understand the landscape of modern Kazakh population. And at the end, I would like to thank all our foreign collaborators and local collaborators, so Professor Lee, Professor Terwilliger, professors from Genomics Institute, Seoul National University, Professor Sayenko, Iragunovich Tatiana from Nagasaki. And of course, I would like to thank laboratory of Professor Akilzhanova, laboratory and the members of Shulpan Askarova and Professor Kushugulova. Then I would like to say special thanks to Professor Rakhbekov Tolibay for his administrative support during our expeditions, and the director of core facility Aidos Balmuratov. And great thanks to every participant and members of our lab. So I'm proud to say that most of the results that we get only using the knowledge of our young researchers. So at the end, I would like to say, as our greatest scientist, Nazarbayev, did before for Kazakh nation in terms of identification and mapping chemical elements under the Kazakh steppe. So today we are trying to decipher the keystones and genetics elements to contribute to better understanding of the landscape of Kazakh nature using genomic technologies. Thank you.
Thank you very much, Polygenic. Do you have questions? We have time for one, maybe, question. Or later we can discuss during lunch and during breaks and during our reception. Thank you very much. A lot of work. Thank you, Polygenic.
Thank you.
Yes. I would like to present next our speaker. He is also representing of our sponsor companies. We say very great thank to our all sponsors today who give also a chance to have this event. And Dr. Krzysztof Barkowski, microscopic imaging system specialist, evidence scientific. Evident confocal systems, new horizons in microscopic imaging. Today afternoon we will have master class. Please, who will be interested, pay attention and take part on this master class. You have microphone there. Yeah, thank you. Company VisaMed.
Здравствуйте. Доброе утро. Хорошо. Dzień dobry. I'm from Poland, so I understand Russian, so this is the reason that I try to use some words. Thank you very much that I can spend with you next 15 minutes. I will try to do it as fast as is possible and as good as is possible. So the title of my presentation is Evident Confocal System, New Horizons in Microscopic Imaging. First, I would like to say, what does it mean evident? Because maybe you don't know, but 3 years ago, company Olympus decided to split from the organization. It was called Olympus Scientific Solutions Division. From this time, we are evidence scientific company, and we are the only producer and developer of Olympus microscopes. So this means that if you want to buy confocal microscope, if you want to other microscope, Olympus microscope, you have to buy it from Evident company in Kazakhstan from VisaMed. Please join us on our booth, we will show you something more. So today I would like to show you some nice pictures, because I'm a microscopist. I liked my job, and so my job is to do as much nice pictures as is possible. So this is the reason that, but today I would like to focus on the special technique which is called confocal microscopy. This technique is really old, yeah, because it was invented in 1957 by Marvin Minsky. It's good to know that there is something which is older than me. Thanks. And I would like to explain why confocal microscopy was invented. Because if you work with the, I will say, use the name ordinary microscope, you are using a wide field illumination. Wide field illumination is really very nice technique, but you can observe, especially if you work with fluorescence, not only images from the focal planes, but also images from unfocal planes. So you can observe only a very blurry image. In confocal microscopy, it's done on this way that you can observe only this focal plane image. It's really easy if we try to understand that we can do this. And it's really nice image because the rest of focal planes are excluded from the observation field. But how it works? Yeah, the idea of Marvin Minsky was to find a hole, a very small hole. Very small hole which let pass through only what is required. So this means that, and Marvin Minsky found this hole. In confocal microscopy, it's called pinhole. And we have now two techniques. One way, but two different methods to exclude these unfocal planes from our image. One is called point scanning microscopy. The second is called spinning disk confocal. From the beginning, we have to use this small pinhole to use it. On the point scanning microscopy, the system of galvanometric mirrors are moving the laser light on our specimen. It doesn't depend. It can be a specimen mounted fixed on the glass slide on the cell vessel, and the light, the emitted light from our specimen is going through this pinhole, so we can observe plane by plane. Because the main idea of Marvin Minsky was to find the microscope for 3D reconstruction of brain structure. On the other side, we have spinning disk confocal. In spinning disk confocal, we are using a rotating disk. But this rotating disk has also a pinholes. This is really the same techniques. And this is the basic technique for the confocal microscopy. By the way, the first spinning disk was invented by Nipkow. It was dedicated for TV. It was the first version of TV. In the modern time, we are using a special spinning disk which are the set of two disks. One disk is with the micro lenses to increase the light intensity, and one disk is with the pinholes. So we have two ways, and how to choose the right solution for right application. Yeah. Why confocal laser scanning microscope? Because of the high optical resolution. This microscope gives you an opportunity to reach the higher resolution than the normal optical microscope. Because we can eliminate light coming from outside, and we can focus on the focal plane, and we can use different light sources. Of course, I mean different wavelengths. Why confocal laser scanning microscope? Because of the sensitivity and fluorescence signal. We can use a longer exposition, and we can find a better signal for very, very faint samples. The next reason that we are using a confocal microscope is high dynamic detection. Because using the confocal microscope, we can acquire weak and strong signal on the same image. It's really very, very nice feature. So, if you need confocal microscope, you have to start thinking what kind of specimen do you have? What kind of sample do you have? And we can offer you system based on the inverted microscope dedicated for live cell imaging, system based on the upright microscope dedicated for the typical glass slides. It's dedicated for the documentation, and upright microscope frame dedicated for electrophysiology. So it depends on you, depends of the application that you are going to use. And this is an example how works scanning confocal microscope and how works spinning disk confocal microscope. Because in scanning confocal microscope, the image must be scanned point by point. In the modern confocal microscope, you can reach at least resolution 4K, so this means that these points, the number of these points is really huge. So the main difference is the speed, scanning speed. Yeah, in the modern confocal microscope, laser spinning scanning confocal microscope, we can reach the speed 500 frames per second in comparison to the best laser scanning confocal microscope, 30 frames seconds. So every time if you think about acquiring the live processes, very fast processes, you have to switch your thinking to the laser spinning confocal microscope. And yeah, why do we have to use this spinning confocal microscope? For imaging large multidimensional areas very, very quickly. This is a nice example of the whole mouse embryo. For capturing very fast cellular dynamic processes. As you see, we can reach almost the same resolution that we can reach using the laser scanning confocal microscope, but we can do the very fast acquisition. We can add a typical resolution, maximum resolution for the confocal microscope is about 260 nanometers. But using additional equipment, we can reach a super resolution. This means 120 nanometers. It doesn't depend what kind of microscope can we use, will we use. And why we use a spinning microscope when cell health is priority. Because if we work with live cells, phototoxicity can kill our specimen. So this is the reason that we have to use this kind of spinning disk microscope. Yeah, and our family of confocal microscope based on the spinning disk technique is always based on the inverted microscope for live cell imaging. We can offer 3 versions of this system, one based on the Yokogawa disk, one based on the Crest Optics disk, and system SR mean super resolution system with higher resolution than a normal confocal microscope. Every time when I discuss with our customer what is the most important part of the microscopy, I have to say the specimen. The next part is the objective. So we can offer every kind of objective, including dedicated silicon objective. Silicon immersion is required when we work with the live cells, because silicon objectives offers us the same refraction index. So this means we can avoid the spherical aberration. We can offer also a water immersion objectives, and so this is an example that how works water immersion objective, silicon objective, and typical oil immersion objective. Last time, this year, we also launched universal objective, which works with silicon and water immersion, but this universal objective has also a possibility to work with silicon pad. So this means we can put on the top of the objective a pad, silicon pad. We don't need to exchange the oil immersion during the image acquisition. It's really very critical during the long time experiments. This is an example what kind of images can we acquire using this kind of objective. As I said, the most important part of our system is the optics. Olympus is known as a company who can deliver the best optics on the world. We also launched a new inverted microscope, which is the base for our confocal systems. It's called AX85. The unique feature for this microscope is the biggest field of view, 26.5. So this means that we can scan much faster because we have to, acquire less number of pictures. Thank you very much.
Thank you very much for interesting talk. Of course, microscopy is also a very important part, not only genomics, not only sequencing platforms. And today we will have special masterclass. Please come and see. It will be in S1 block in our Center for Life Science building. in 1 p.m., I think, 1:30 p.m. will begin. Please see techniques and what will be specific for microscopy. Thank you very much for your talk. And also, I would like to say one advertisement about our poster session. Poster session and poster exhibition take place in the upper atrium. You can find on the right way after going out from this ballroom. Posters will be 3 days session, and on the third day we will have a poster contest who will be best from this poster session. And on the third day also will be who will be best oral young scientist talk. But today you can see posters and evaluate, and today you can see also exhibition of our sponsors. Thank you very much, and we will have break for lunch. And we will wait here at 2:15. Thank you.
Ma io gliela do tanto come messo perché io
Раз, чек. Раз. Раз, чек.